Good evening, everyone. I hope you're doing well. And let's dive right into our next lecture on biological resources. And this is the first of two lectures on biological resources where we talk about the life aspect of these resources. And here's another picture of that deer from Yosemite National Park. Here are some more deer from Yosemite National Park up in the Tuolumne Meadows area. And this is the species Otocoileus hemionus, the mule deer, and that is the species name for these organisms. And I want to go back to what we've talked about earlier in the semester, exactly what is a species. And when you look at these deer, obviously they are a species. And as we've discussed previously, a species is traditionally defined as a group of organisms that can successfully reproduce amongst themselves, but not with any other organisms. And as we've discussed, in other words, a species is reproductively isolated from all other organisms. And this concept of reproductive isolation defining species is known as the biological species concept, which is often abbreviated by biologists as the BSC. And the biological species concept has its basis in what's known as Linnaean taxonomy, which was developed in the 1700s by Carolus Linnaeus and is still in use by many biologists and scientists today, where a given species will have a given name and then it fits within a ranked hierarchy. So, for example, humans are in the kingdom Animalia, the phylum Chordata, the class Mammalia, the order Primates, the family Hominidae, the genus Homo. Our species is Homo sapiens, and then we are actually considered to be in a subspecies known as Homo sapiens sapiens. I want to look at that species name. It's what's known as a binomial name, which means you have to have both parts of that name. Homo sapiens are always listed in the species, and the genus and species names are also always italicized. So this would be the hierarchy for our species, Homo sapiens, but you can do the same thing with any other organism. For example, here you have the same hierarchy for horses, where they are in the order Perissodactyla, the family Equidae, the genus Equus. The species is Equus caballus, and again, there's that binomial. And then the subspecies of domestic horse is known as Equus caballus caballus. And so this is the traditional way of organizing different organisms, naming them, and then putting them in various categories in a growing hierarchy that relates them to other organisms. There are some exceptions to this rule. For example, this is a mule, and he doesn't have a species name because mules are not a species. This mule, back in the days when they used to have mule rides up in Yosemite National Park, his name was Guinness, and it's an awesome name for a really, really nice mule. And a mule is a hybrid. A mule is a hybrid of a female horse and a male donkey. And when you have a male horse and a female horse and a male donkey get together, they can reproduce mules. They can generate living organisms because horses and donkeys are actually relatively closely related. As far as we can tell genetically, they only separated as different species from each other about 4 to 4.5 million years ago, which sounds like a long time. But since horses have long generation times and only have one foal per birth, generally speaking, it takes a long time for speciation to take place. And so these animals are closely related. So closely related that they can give birth to live young that then survives. However, the distinction why horses, how we know that horses, one of the ways that we know horses and asses or horses and donkeys are separate species is that their offspring, mules, are sterile. So that is also an important part of the biological species concept, that you have organisms that can reproduce. If you are in the same species, you are reproductively isolated. You can only reproduce within your own species, and your offspring have to be viable and fertile. And so if you have infertile offspring like mules, then technically you are not in the same species. And so horses and donkeys are different species. They're closely related enough that they can produce viable offspring, but not fertile offspring. And even then, occasionally they can. I think one in every 200,000 offspring you'll get a fertile mule, but it's an extremely rare event, and that shows the distinction between horses and donkeys as species. Now, in terms of species, I want you to take a break from thinking about mules and donkeys and horses and think about yourself and where you are in your life right now, and all of the different things that went into you being here, all the places you've been, all the people you've met, all the decisions you've made. Think about all of that. And then put yourself mentally about 10 or 15 years earlier than you are now, looking forward. And how many of those choices you made, how many of those places you went, how many of those people you met did you plan on? How many did you anticipate? How much of where you are in your life right now was all mapped out by you 10 or 15 years ago? And if you're like most people, a lot of your life was planned, but a lot of it was accidental. You just happened to go somewhere and meet somebody. They happen to make you an offer. You happen to meet a significant other, something like that, that changed your life. And that led you to being where you are today. And so that happens to so many of us. It certainly happened to me, and I think it happens to most people. And the same thing is true for species. We've already had a previous lecture where we talk about how species form and how speciation takes place and how evolution is documented. Where if you have this being the distribution of a species, and suddenly you have a population that gets separated from the rest of the species, if that separation prevents gene flow between the population and the rest of the species from taking place, then if that happens long enough, that population can potentially become a separate species. That's what happened with donkeys and horses. There was a separation 4 to 4.5 million years ago, and they remained separate long enough, and gene flow was prevented from crossing between them long enough that they eventually became different species. Even though today humans can breed them to produce mules, nevertheless, in nature, that sort of thing doesn't happen, and they remain separate species. But what happens if that barrier between the population and the rest of the species comes down before the population has genetically diverged enough to be a separate species? If that happens, the barrier comes down, gene flow is restored, and as a consequence, you're back to just having that same species that you started with. And so right now, predicting new species would be very difficult because it's entirely possible, if we look at any living species today, that there is some population out there that is geographically isolated or isolated in some other way from the rest of the species, and there's no gene flow taking place. But predicting whether that will remain the case for another 10 years or 100 years or 1,000 years or 4 to 4.5 million years, we don't know. We can't predict that far in advance. And so species are difficult to predict in advance. It's hard to say that this organism is eventually going to be a different species from the parent organism. And that's why species are best thought of as something you see with hindsight, like a bison in your rearview mirror. We can look backwards and see all the things that led this species to diverge from other earlier species. The same as you can look at your past life and see all of the choices that you made that led you to be where you are now. But you can't necessarily predict those in advance, just as you don't necessarily know where you will be in your life in another 20, 30, or 40 years. And so thinking about species in this way, you realize that it's really almost impossible to predict what species will be with us in the future, even if those species are forming today. We only see those species in hindsight. And yes, as an aside, I'm perfectly aware that as a paleontologist, this would have been a much more appropriate rear view mirror image. And yes, you're right, but this is also too easy. It's such a cliche now. I'm just not going to go there. If you think back to our lecture on evolution, and we talked about the modern evolutionary synthesis, well, this gentleman here, Ernst Mayr, is the one who first proposed the biological species concept, and he's the one who came up with it. And just as another aside, I actually got to meet him and have lunch with him. He came to give a lecture at UC Riverside back in the 1990s, and I got to meet him. And some colleagues and I went out to lunch with him, and he had beer and he had bratwurst, and he was a very, very nice guy, even though he was in his 90s by that point in time. And we talked about things like the biological species concept, and we talked about fossils, and we talked about hominins and so on. One thing I wish I had asked him, though, was about this critter. This is, as you probably know, this is an amoeba. An amoeba are single-celled organisms. They are just a single cell, and they reproduce by fissioning. We talked about that sort of reproduction when we talked about viruses in an earlier lecture. Well, the same thing is true of amoebas. They reproduce by fissioning. They reproduce asexually, which means they don't have sexual intercourse with another organism. They simply reproduce on their own. And if that's the case, if they don't need a female or an opposite sex in order to reproduce, then how do you know that they are reproductively isolated from other species? Granted, they are. You can tell that an amoeba is not having relations with other organisms, but still, how does that work? How do you draw that line? It's very difficult with asexual organisms. It's also a problem for those of us who work with fossils. Here are a couple of fossil horse skulls from the ice ages of North America. The top one from southern Nevada outside of Las Vegas, and the bottom one from Texas. Are these the same species? I can't use the biological species concept to tell. I could put both of these skulls in a cabinet. I could turn the lights down low. I could play some romantic music, and absolutely nothing is going to happen. I open that cabinet the next morning, and I'm still going to have two horse skulls sitting there. I can't demonstrate with fossils whether or not they were reproductively compatible. And so I can't use the biological species concept for fossils. Even larger than microscopic organisms have this issue. If you look at things like ferns that simply release spores that then plant themselves, it's also asexual reproduction. And so there are a variety of organisms for which the biological species concept doesn't quite fit. And so the biological species concept does not always work effectively for fossils or for asexual organisms. And so, what other options are there for us to figure out what is a species? Well, let's look at these animals. As you probably know, these are zebras. In zebras, you just look at them and you know that they are something unique and special. And you can also look at them and compare them to other organisms you're familiar with. And just by a process of elimination, you can say, well, you know, zebras are more closely related to horses than they are to anything else because they just look like horses with stripes on them. So you could call them horses, except that they aren't horses because they have those stripes. And so this is back when people were first when natural philosophers were looking at dividing the natural world into different groups of organisms. It was easy to unite zebras with horses, but separate them out as a distinct species because of their striped pattern. And so it seemed very, very clear, very reasonable. So what would you do with this organism? Here you got something that looks like a zebra at the front end and looks like a horse at the back end. And in fact, it doesn't even look that much like a zebra at the front end because instead of black stripes on a white background, you've got dark brown stripes on caramel or cream colored background. So kind of zebra-like in the front, not at all zebra-like in the back. What do you do with this? What do you relate it to? Well, in case you didn't know, you should know, but in case you don't, this is an animal known as a quagga. And I love quaggas. I think they're just some of the coolest animals out there, or at least they were. Quaggas used to be interpreted to be one of four basic kinds of zebras that were out there. You had quaggas, you had plain zebras, you had mountain zebras, and then you had grevy zebras. And each of these zebras could be distinguished based on their coat pattern, how big their stripes were, how fat their stripes were, how many stripes they had, where their stripes were, how far down their legs their stripes go, and their body size. All of those were characteristics that were used to define different kinds of zebras. And the other distinguishing feature about quaggas is that they are extinct. We don't have quaggas alive today. That's why I'm showing you old pictures of them. The last quagga died in the Amsterdam Zoo back in 1883. And this is a picture of, I believe this is actually a quagga in the London Zoo before it died, but this is what a quagga looked like in life. And the reason I'm going off on quaggas is A, because I like them, and B, because quaggas give a nice example of what's known as the morphological species concept. This is different from the biological species concept because here you're not using reproductive isolation at all. This is strictly looking at the morphology of the organism. And when we talk about morphology, it's a study of the form and or structure of things. So when you use the stripe pattern, the coat pattern, the body size, the body build, and so forth, to distinguish one kind of organism from another, you're using the morphological species concept. And quaggas are a great example of this. Now, the thing is, I want you, number one, just as yet another aside, look at the proportions of this quagga. This is a photograph, and so this is what quaggas look like. And you can see it's got very zebra-like proportions with a short, stocky body and a great big head on the front end. And yet when artists painted quaggas back in the 1700s and 1800s, they painted them, not just this guy, but all sorts of them, as very horse-like, with big horse-like bodies and horse-like proportions and relatively smaller heads at the front end. I have no idea why that is. I just think it's really curious and kind of fun. This particular quagga painting, you can see a lot of French writing down at the bottom, and this is because this was a fairly special quagga. This is a quagga that was brought from South Africa, where these animals lived, and brought to Versailles in France, where it was living alongside King Louis and Marie Antoinette. They saw this quagga. They had quaggas and other wonderful, wild, and at the time, virtually unknown organisms brought back to live with them in Versailles, one of the perks of being royalty. One of the downsides of being King Louis or Marie Antoinette was the French Revolution. And when both of them lost their lives and were beheaded, this guy, Jean-Baptiste Lamarck, who you may remember from our evolution discussion, he was one of the precursors to Charles Darwin and Alfred Russel Wallace. Lamarck is the one who proposed the idea of inheritance of acquired characters. Well, he was also working for, or he was employed by the French government. And so when the French Revolution happened, he removed the quaggas and other rare and special animals from Versailles and moved them to the Royal Garden. And since royal anything was bad during the French Revolution, the garden was renamed to just the Plant Garden, which is still present in Paris today. It's the Jardin des Plantes. And you can go there today. And the quaggas made it there and survived there and lived out their lives there. So if you go to the Jardin today, this is what it looks like. There's your statue of Lamarck sitting there prominently displayed. And in the background of this picture is the Natural History Museum in Paris. And if you go to that and pay a few euros and go inside, you can see that same quagga. It was preserved. So this quagga lived in South Africa. It was carried from South Africa to Versailles, where it was enjoyed by King Louis and Marie Antoinette, saved by Lamarck, moved to the Jardin, and when it died, its remains were preserved, and it remains in the same Jardin even today, where you can go see it. And this is not just a historical oddity. This is not me encouraging you to go on a tourist trip. The fact that this quagga is preserved means that you can actually sample its tissues for DNA. And so you can sample DNA and compare this quagga with living zebras. This has actually been done. And what's been determined based on genetics is that quaggas are actually plains zebras. They're just a regional variation of plains zebras, which means quaggas are not technically extinct. It was a regional extirpation of a kind of plains zebras, because plains zebras are still with us today. In fact, they're one of the few kinds of zebras that is actually relatively healthy. They aren't vulnerable. They aren't threatened. Their populations are doing relatively well. And so quaggas were just a local elimination of a kind of plain zebras. I hope you were writing that down, and I hope you were paying attention, because this is going to be on the test, and I swear I'm going to give you the answer. If I use the question is going to be true or false. A quagga was an extinct kind of zebra. And I just told you that it wasn't an extinct kind of zebra. I told you that it was a plain zebra and it was regionally extirpated, and that's it. And the reason I bring this up is that last quarter, I had that same question on the exam. True or false? A quagga is an extinct species of zebra. 98% of the students picked true. It's not true. It's false. Quaggas are not extinct. Quaggas are plain zebras, and plain zebras are doing relatively well. So if I ask that question, true or false, a quagga is an extinct species of zebra, the answer is false, because their relatives, their same species, plain zebras, are still very much with us. I'm sitting here giving you answers to the exam. How nice is that? I hope you appreciate it. The other thing I want you to take from this discussion of quaggas is that the morphological species concept does not necessarily reflect true species relationships. Even though quaggas look very different from plain zebras, even though you can tell them at a glance because one looks like a zebra and the other one looks like a zebra up front but a horse in the back, nevertheless, they are the same species. And so the anatomical characteristics, the morphological characteristics that you're using to distinguish between one or the other of them don't reflect the actual relationship between them. And so the morphological species concept also has issues. So now we've talked about two kinds of species definitions, the biological species concept and the morphological species concept, and both of them have some challenges. What else can we use? Well, as we've just discussed, there's the genetic species concept, where you use genetic material, DNA, to determine the relationship between different kinds of organisms. That's how we know that quaggas are actually just plain zebras. And so this would appear to be a very useful tool. Let's go back to these horses. In terms of horses, in terms of fossil horses, I've already explained why the biological species concept doesn't work, because fossils don't reproduce. But as a paleontologist, I specialize in anatomy, in morphology. And because these are horses, these are my particular specialty. So these are the organisms that I focus on. So if you had asked me prior to about 2014, 2015, how many species of Ice Age horse there are in North America, I would have listed these and shown you the characters that I use to distinguish among these different species, and then applying the names to the different groups that I see. And so you've got 9 different kinds of Ice Age horse here. This is based on morphology. This is based on the anatomy of the fossils that I've been studying my whole life. So imagine how I feel as a scientist when in 2017 there is this paper that takes all of those horses I just showed you and puts them in one group. It says that where they can get genetic material, the genetic material doesn't show any differences. So what I'm looking at, whatever it is, isn't species relationships. It's something else. And if you want to know what it is, so do I, because I haven't figured that out yet. But this is what caught me by surprise. It was a little depressing to have spent your entire life looking at fossil horses, breaking them out anatomically, and then just in the past 5-6 years, having genetics specialists come in and look at and sample these fossils for DNA and look at the genome of these fossils and determine that, to the best of their ability, all of these are one species. Coming back to this skull, this is the same skull I showed you earlier. That is my colleague Christina Lutz, who is working with me on some of these horses from Nevada. And the reason I show you this skull again, number one, is because we were very happy to find it. It was originally discovered in a cave outside of Las Vegas back in the 1930s. It went to the LA County Museum in the 1950s, and it disappeared. And it wasn't until just a couple years ago that we relocated this skull in the collections. And it hadn't been lost. It had been moved to the modern bone collections to be used for comparison because the preservation is so good. In this particular cave outside of Las Vegas, the fossils were mummified due to the dryness and due to the heat. And as a consequence, this looks like it's a horse from historic times. In fact, somebody even wrote burro or wild ass on the top of this, and then they put it in the modern bone collections rather than in the fossil collections. And so that's how good the preservation is. But Christina and I recognized that this was actually a fossil, and we collected material. We sampled this skull. That's how we know it's a fossil. We dated the fossil, and it's more than 13,000 years old. But because the preservation is so good, we were also able to get DNA out of it. And the DNA was shared with my colleagues Pete Heinzman and Beth Shapiro. You are not going to be expected to remember their names. I'm just sharing their names with you so that you don't think this is something that I'm doing all on my own. I don't do DNA. I just cut holes in the bone and sent the samples to these guys up at UC Santa Cruz. And this comes back to their phylogeny that was published back in 2017. And here are all of the horses I mentioned earlier, all of those Ice Age horses from North America, plus living domestic horses, living feral horses, plus living zebras, plus living asses. All of them fall in that box. And the skull I just showed you is completely separate. Even though it looks like a horse, even though it looks so much like a horse that somebody labeled it burro or wild ass, nevertheless, it is a species of horse that's separated from all other horses more than 5 million years ago. And you would never be able to tell that from the morphology. You needed the DNA in order to determine that. And so that's why the genetic species concept can be so powerful, not just for recently lost organisms like quaggas, but even going back thousands of years into the fossil record. You can use the genetic species concept to look at the DNA of ancient organisms and figure out their relationships. I will mention that I do have some colleagues who didn't like this interpretation from 2017. They essentially ignored it and went back to using anatomy, to using morphology, because what they thought they were seeing in the fossils didn't match what they were seeing in the genetics. I have a problem with this, even though I know each and every one of these authors and I consider many of them to be friends. I still have a challenge with this. If you go back to one of our first lectures where we talk about the nature of science and the different things that science has to be, one of them was that science is supposed to be comprehensive, which means you take all of the data into account. You aren't supposed to leave some data aside and say, we don't agree with it, so we're going to leave it behind. You don't do that. So this paper that was published in 2019, I have some challenges with. You don't get to ignore the genetic data and focus just on the morphology, especially since we know that sometimes the morphological species concept gives you a false signal. I find it frustrating because if you remember our evolution lecture, I talked about, and many other scientists, paleontologists, biologists, evolutionary biologists, anthropologists, all sorts of people talk about how you can use DNA to link all living organisms. And this is amazing proof of the theory of evolution initially proposed by Charles Darwin and Alfred Russel Wallace, and eventually refined in the modern evolutionary synthesis. None of those people had DNA available to them to study. And so when DNA becomes available and people start collecting it and sampling it and studying it and seeing those same relationships represented in DNA, it is a stunning proof of the concept of evolution. And it's particularly powerful because it's independent. And so all other paleontologists, or at least competent paleontologists, they will rave about this connectivity and the importance of DNA in demonstrating the validity of evolutionary theory. And yet when you get down into the nitty gritty about fossil horses, they will say, oh no, those DNA people, they probably don't have a complete sample of the genome. They probably missed something. They're probably using bad characters. I'm going to stick with the anatomy. I'm going to stick with the morphology. You don't get to do that. And I'm sidetracking it like this because I want you to continue to remember that science is a process and that scientists are human and that sometimes it takes a while for the actual answer to emerge. What is the relationship of these animals? We're still working on it. We're still figuring it out. I personally think there's something we're all missing here. And it might take another generation or two and much better minds than mine to figure it out. But I'm emphasizing that this is the case even when you have genetic material. There is still some discussion, there's still some examination of the data, and scientists still get involved and sometimes even argumentative about this sort of thing. And all of this misses the point. What happens when you can't find genetic information? This is the lower jaw of Lucy, the hominin fossil that we discussed earlier. I hope you appreciate that I got a close-up view of Lucy's jaw for you, because most people don't get to see Lucy up close like this. But her jaw is so mineralized, it is so fossilized that there is no organic material left, which means you cannot get DNA out of this fossil. There's simply no way of doing it. And as a consequence, you cannot use the genetic species concept to determine what Lucy's relationship is to other hominins and to ourselves. It's not an option to you because you need that genetic material in order for that to take place. And what's true for Lucy, who's only a little more than 3 million years old, is certainly true for organisms like Tyrannosaurus rex, who goes back 66 million years and more. You might find some exquisitely preserved remains that have trace fragments of DNA preserved. I don't think that has happened yet, but nevertheless, it might happen. But that won't be enough to reconstruct a Tyrannosaur genome. So you won't be able to relate Tyrannosaurus to other dinosaurs or to birds or to other organisms. And so for these organisms, the genetic species concept simply doesn't work. And so the genetic species concept requires relatively intact DNA sequences, and these are not always possible to obtain. And so here is a challenge with the genetic species concept. Despite its power, despite how useful it is, it nevertheless does have some challenges. A lot of the horses that I've studied that I showed you earlier in that list don't have DNA in them. So the anatomy is all that you have to go on. The morphology is all that there is. I'm just not sure it's being interpreted correctly. That's why I think there's still more room for exploration in those studies. Other kinds of species concept, there's the phylogenetic species concept. And this is where you take different groups of organisms and you link them based on shared derived characters. So here, if you look at just this group of organisms, you have the appearance of mammary glands separates a group of organisms from non-mammalian vertebrates. And then within that smaller group, some of them are live-bearing. That is a derived trait. And so that separates those from egg-laying mammals. And then you have organisms like us with a placenta, and that separates us from things like marsupials. And so it's a way of establishing a branching relationship where you don't use all of the anatomical features of an organism. You just use shared derived features in order to link organisms in increasingly small branches. And so, for example, here's a cladogram of the dinosaurs, and this is exactly what they've done with various anatomical characters. I emphasize that this uses anatomical characters the same as the morphological species concept does. The phylogenetic species concept uses anatomy to link organisms into branches, or in this sense, what are known as clades. That's why another name for the phylogenetic species concept would be cladistics. And so you have these different clades of dinosaurs all branching out, and they're linked by shared derived characters. Are there challenges with this? You had to have seen this coming. Yes, there are challenges with this. For example, here's an older cladogram. This is from 1980, and this gets back to Ice Age horses. And this was a cladistic analysis of Equus. Equus being the genus of horses. And in this case, they use living horses as well as fossil horses. And what I want to point out is here you've got quaggas, and you can see that they are the end of that particular branch or that particular clade. And yet here is Equus burkeli, which back in 1980 was the scientific name for the plains zebra. And we just showed genetically that plain zebras and quaggas are the same species. And yet here in this cladogram, they're on completely different clades. They're on completely different branching lineages. And the implication from this cladogram is that they're completely different animals. And we know that that's wrong because genetically we know that quaggas and plain zebras are the same species. And so here's another indication that what we can learn from genetics isn't necessarily reflected in the morphology. And so characters selected for use in phylogenetic analyses may not always necessarily reflect true species relationships. And so yet again, you have another challenge with this particular way of defining species. So what is a species? I'm sure you're very frustrated at this point. How do scientists figure out what a species actually is? And the answer is scientists are still discussing this. Scientists who use the phylogenetic species analysis have a different take on things from, they don't even use they'll use the binomial naming system of Linnaeus, but they don't use that kingdom, phylum, class, order hierarchy that Linnaean taxonomy employs. They use branching relationships. So when you talk to somebody doing phylogenetic species analyses, they're going to define species differently than somebody who is using the Linnaean hierarchy. And both of them are going to define species differently from somebody using the genetic species concept. And so it can be very frustrating. And sometimes scientists will say, I am looking at this species, and they don't tell you how they know. And it can be very frustrating for the uninitiated. If you just hear a scientist saying he or she is talking about a given species, unless they tell you how that species definition was arrived at, it's not necessarily helpful. And even the concept of species itself is coming under discussion by scientists, where increasingly many scientists are looking at things like the biological species concept and saying, you know, in many cases, it doesn't work. There are organisms that we consider to be different species that nevertheless are capable of breeding with what we would consider to be other species. For example, bison can reproduce with cattle and produce offspring. And that's not supposed to happen necessarily if they're different species, and yet clearly it does. And so as we look more and more at the natural world, increasingly it looks like we as scientists keep trying to put things in boxes and put things within parameters. And the natural world keeps saying, not so fast. I'm more complicated than that. There's more to it than that. And this is among many of my colleagues a relatively recent realization, but it actually goes back much further. You can actually find musings about this in books like this. And I strongly encourage you in your spare time to read this book because I think you'll have a lot of fun. This is the log from the Sea of Cortez. And in this book, which is relating a voyage that was made to the Sea of Cortez by John Steinbeck and his friend, the biologist Ed Ricketts, back in 1939 and 1940, there is this quote. Indeed, as one watches the little animals, because they're looking for animals in tide pools and collecting them and cataloging them and categorizing them, as one watches the little animals, definite words describing them are likely to grow hazy and less definite. And as species merge into species, the whole idea of definite independent species begins to waver, and a scale-like concept of animal variation comes to take its place. And so more than 50 years ago, 60 years ago, on the Sea of Cortez, you had these people looking at living biological organisms, and the more of them they looked at, the more they realized that species definitions are really inadequate for actually capturing the number of different life forms that are out there. And so that is important to keep in mind as well, that as scientists continue to explore and explain and categorize the natural world, we're still figuring out what the best way is to do that. So if we talk about a species, realize that a species is a concept imposed on nature by scientists, and that sometimes nature can be a little more complicated than that. With that in mind, how many species are there on the planet? Here again, because people define species differently, and because there are different ways of defining species, and because species definitions have been promoted for decades and centuries, there is discussion and disagreement about how many species are out there. For example, here you can see a study in 2011 estimated that there were 8.7 million species on Earth. A study in 2013 said that there were 5 million species with an error factor of plus or minus 3 million species. That's a huge error factor, by the way. And to give you another answer on the exam, I might ask you, whether an investigation in 2013 estimated 5 million species plus or minus 3 million species, or I might ask you true or false. A study in 2013 estimated between 2 and 8 million species. It's saying exactly the same thing. It's just saying it differently. So if you get that question on your next midterm or on the final, please keep that in mind. A still more recent 2017 study proposed anywhere from almost 1.5 billion with a B species to over 5 billion species. And this was distinct because it included bacteria and parasite richness, which hadn't been done in those earlier studies. So scientists are still debating how many species are out there. The one number I would like you to remember is that there are roughly 1.7 million named species on Earth. These are species that scientists have actually given a name to. And if we've got more than 5 million or more than 8 million or more than 5 billion species out there, it means that there are a lot more species out there remaining to be discovered and described and named. So paleontologists as well as biologists have their work cut out for them in finding, discovering, and naming these previously unrecognized organisms. And at this point, you're probably wondering, why do we care? Why is it so important that we figure out how many species there are? Well, there's a variety of reasons. Number one, all of your food comes from other organisms. Everything you have ever eaten in your life, except that one time you were a little kid and you tried eating dirt, everything you have eaten in your life is either another organism or it's produced from another organism. And we had our lecture on agriculture, and we had our lecture on how we eat as Westerners, as Americans, we eat relatively few other organisms, but still keeping a handle on how many organisms are out there, how many of them are potential food sources, how many of them are part of ecosystems that support the food sources that we do utilize. All of that is important for determining whether or not given communities, given ecosystems are healthy. And so just in terms of our own self-interest, in terms of what we eat, it's important to know how many other species are out there. It's also important for things like medicine. Many medicines are based on other living organisms. In fact, more than half of all modern medicines are derived from or modeled on natural compounds from wild species. And so medicinally, other organisms are very important. In fact, it is a major endeavor of biology today to go out into remote areas and look for new species and determine whether those species might have medicinal properties that people can use. That's a huge driving thing in biology today. Other organisms, as I've just mentioned, are already important for their ecological significance. Organisms can help transform and maintain ecosystems. When you talk about agriculture and the need to pollinate plants in order for them to reproduce, you depend upon biological processes in order for that to happen. The organisms are also necessary for soil formation or for waste disposal, purifying air and water so that we can consume it, for cycling nutrients, and obviously for absorbing solar energy to support the trophic levels of which we are a part. So for all of these reasons, species are important. And so it's important to know how many species are out there and how they relate to one another. And finally, of course, there's just the aesthetics of other species. So much of what we consider beautiful on our planet is other living things. And so to maintain the health of that beauty, it's important to know what species are involved in producing that beauty. Which brings us to the idea of biodiversity, determining how diverse life is on the planet. And when you're measuring biodiversity, there's a couple of different ways I want you to keep in mind. Number one is species diversity, which is very easy. How many different species are there within a given community or a given ecosystem? And then there's genetic diversity. And genetic diversity is the variety of different versions of the same genes within a species. And here I am being nice again, because I usually ask a true or false question about this where I reverse the definition. So true or false, species diversity is the variety of different versions of the same genes within a species. And that's obviously not the case. That would be the wrong definition. So if I ask you that as a true or false, make sure you're reading it correctly and make sure you know the definitions. Within species diversity, you also have species richness, which is the total number of species in a given community, and species evenness, which is the relative abundance of individuals within each species in a community. How many, for example, in Yellowstone, shown here at the bottom, how many bison do you have in an area? How many pronghorn do you have in an area? How many elk do you have in an area? How many wolves? How many bears? What is the relative abundance of those organisms? That is species evenness, which is different from the total number of organisms in that community, which would be species richness. So please keep those terms and their definitions in mind. And with that, I'm going to go ahead and end the lecture here. Our next lecture is going to be on the other side of life, which would be death and, more appropriately, extinction. So until then, I hope you're doing well. Please take care. Please stay safe and stay healthy. And we'll talk to you down the road. Have a good evening.